We have one last environment for this class for this semester, and that is the air environment. So in this pre-lecture, we're going to talk about some of the basics of what's going to go on with aeromicrobiology so that you're ready to go when we get in class and discuss some more details. So when we talk about aeromicrobiology, we're talking about what we call bioaerosols. So airborne biological materials are called bioaerosols. That's the terminology that we use for them. And when we look at aeromicrobiology, we're looking at two different places where we could be studying. The first is extramural, and extramural just means outside. So it's related to aerolization of indigenous soil pathogens, pandemics, microbiology in the clouds, agriculture, waste disposal, airborne toxins, those types of things. So it's related to air generally that's outside, that most people are interacting with on a daily basis. Intramural microbiology refers to what's going on inside a building or a specific structure, and it's specific to public health related to those buildings. And it's not just regular buildings, it's also things like hospitals and laboratories in particular. Hospitals and laboratories are going to be a really important one when we think about intramural aeromicrobiology, because those are locations where we're going to have a lot of airborne particles that could be potentially pathogenic, that are biological in nature. So extramural and intramural microbiology are slightly different areas of study. When we talk about aerobiology in general, that is the study of life in air. So it's a very, very big field. So when we talk aeromicrobiology, we're starting to talk about the microorganisms within that study. And in general, it's also related to the study of aerosolization, aerial transmission, and deposition of biological materials. So that's really important too. Once it's in the air, in terms of a particle or a microbe, it doesn't mean it's going to stay in the air. So a lot of times we're worried about how it's moving in the air or how things are being transmitted in the air. And aerobiology intersects with a lot of different fields, so not just environmental engineering, but things like public health, environmental science, industrial and agricultural engineering, even, believe it or not, biological warfare and space exploration, believe it or not. So we do need to know how organisms live in the atmosphere in order to understand maybe what could be on other planets, which is kind of an interesting area of study. So let's talk a little bit more about bioaerosols. So any particles in the air themselves are called aerosols. When they are biological in nature, they are called biological aerosols. And generally, when we think about aerosols or bioaerosols, we're worried about the threat to public health, and we're worried about public health in reference to respiratory intake and the possibility of deposition of those particles in your nasal passages or your bronchial passages, which are down to your lungs. And one of the things that we're worried about is that smaller aerosols tend to travel further into the respiratory system. And if you've taken anything anatomy-wise, in your lungs you have very, very small branch structures called alveoli, which basically are where the surface area and the contact of gases with your blood occurs, and that's how we get oxygen transferred to your blood. And so we're very concerned about particles that can potentially get down into that level of your lungs, because then it starts to affect your respiratory ability to circulate oxygen in your system. So that's really going to be important. That's why we care so much about small particles. So when we talk about particles, there are two sizes that we are concerned about as environmental engineers. And if you look up air quality standards, these are actually the two that are regulated from an air quality standard standpoint. So the initials P and M refer to particulate matter. PM10 refers to particles with diameters less than or equal to 10 micrometers. And PM 2.5 are particles less than or equal to 2.5 micrometers. And what we know from science and from study is that at these size ranges, we start to see changes in the physiological response to those types of particles. So that's going to be really important. And what you see on the screen for an equation is a relationship between the aerodynamic particle diameter and things like Stokes' law, which we'll talk about in class, and the density of the particle and the density of water. And so atmospheric particulate concentration is expressed normally in units of micrograms per cubic meter of air. And so many times we want to know how big those particles are and how many of them are in a certain volume of air. And the reason that we care about particulate matter is because it's not only something health-wise we should be aware of, but they could also shield microorganisms and be a way of transporting microorganisms. So when we see people inhaling particulate matter, one of the things that we're a little bit worried about is that many times at very high inhalation rates or very high pollution rates, we're seeing decreases in pulmonary function, so pulmonary being your lungs. We're seeing chronic coughs, bronchitis, asthma attacks, things like that. And these can be big. They're not just isolated. So if you think back to we've had forest fires a couple summers here in Nebraska, and we've had very hazy days. That's all particulate matter, and we actually had air quality alerts those days for particulate matter. But you can also see that airborne particles travel great distances. So the satellite photo that's on the right side of this slide is a picture of some intense dust storms that happened in the Gobi Desert between 1998 and 2001. And the Gobi Desert is in western China and Mongolia. And what you're actually seeing is dust over the Sea of Japan. That's how big these storms were. And so airborne particles actually travel great distances, and if microorganisms can attach to those particles, that's going to be of huge importance to us as we're thinking about transport of these particles. So when we think about bioaerosols and we think about all those particles and what we just learned, biological contaminants when we're talking about bioaerosols could be whole entities such as a whole bacteria. We could have viral human pathogens. We could have airborne toxins, dust. All of these that are related to biological components are going to be bioaerosols. And so they can be ingested or inhaled. Inhalation is normally the exposure route that we're most worried about. Ingestion is less likely, but it could happen if air has been deposited on a surface. And when we classify bioaerosols, we use a different size classification. We don't use the particulate matter classification. We have names for these. So when we're classifying bioaerosols, we classify them either as nuclei mode, which are particles smaller than 0.1 micrometers in diameter. We can have accumulation mode, where particles range from 0.1 to 2 micrometers, and then coarse mode, where they're greater than 2 micrometers in diameter. So we've got small, medium, and large particles. And the next slide is going to show us kind of where the range of organisms are going to fall. So when we look at the nuclei mode and the accumulation mode, this is really viruses. Maybe we might have a very, very small bacteria, and it is possible that we would have spores, maybe in this range, depending on the organism. But in general, when we talk about protozoa, fungi, and bacteria, they are going to be coarse particles and in coarse mode. And the reason that that matters is, just like we talked about with soil, these organisms have a great way of attaching to particles and moving with particles. So if we're seeing a lot of particulate matter in the air, there's the possibility that a lot of bacteria associated with those particles are moving, especially in this coarse mode. And so that's going to be extremely relevant to us going forward. The last thing that I want to talk about before we get into class and really discuss this in detail is modes of what we call respiratory transmission. So these are examples for influenza, for the flu, but these are very, very relevant for lots of different diseases. But one of the things that we need to think about when we're thinking about air is, how are these organisms transmitted from place to place and from possibly human to human when it comes to pathogens. So we can have what's called direct contact, which is when transmission occurs when the transmission of the microorganism is direct physical contact between infected or colonized individual and a susceptible host. We can have indirect contact, where transmission occurs by passive transfer from a susceptible host via some kind of an inanimate contaminated object, so maybe particles. We can also have droplet transmission, which is very important. So large droplets that are greater than or equal to about 5 micrometers in diameter come out of the respiratory tract and have water vapor associated with them. And so if you think about coughing or sneezing, when you're coughing and sneezing, you're not just sneezing dry air. You're sneezing a lot of water vapor that contains a lot of those microorganisms. And so you have a lot of mucus, you have a lot of other things that are going on, and the organisms can be in those droplets. And those droplets can be transmitted to somebody. So the droplet mode of transmission is actually the mode of transmission for the coronavirus. And so that's why when we were having the COVID pandemic, everybody needed masks, because masks were not necessarily because of particulate matter, it was because of droplet transfer. And so that's pretty important to us in our lifetime. Then we can also have airborne transfer. So it could be the dissemination of microorganisms by just aerosolizing things that were in a solid or a liquid phase before and bringing them into air. And then the organisms can move around, maybe associated with particles. They could be by themselves, but it's very, very rare. They could be associated with skin cells and all sorts of different types of particles. And they can be very widely dispersed by this mechanism. So these are our different types of transfer when we think about respiratory tract diseases, but they are very relevant to us in terms of thinking about how air is moving, especially between people, because a lot of aeromicrobiology for us as environmental engineers is going to be related to public health and to how buildings are functioning, so intramural microbiology. So hopefully that gives you just a couple terms to get you situated for the class, and we're going to talk in depth about modes of transmission and how aeromicrobiology is going to impact us as environmental engineers.